So previously, we presented the unique ability of our BioDrone technology to produce nanovesicles in a large quantity and its potential as a novel drug delivery system. Starting our journey at the startup CRISPR, we and our sponsor Lonza met together to identify the key objectives to best assess its potential as a drug delivery system, including comparisons between biodrones and exosomes. As previously demonstrated, CDVs can be produced in far greater numbers than exosomes, probably because CDVs are derived from multiple membrane sources. We confirmed this by several means. It turns out that some membrane components are well conserved between the two, while other organelle markers or even some classical exosome markers are overrepresented in CDVs. For instance, some membrane proteins like CD63 are enriched in CDVs, while CD9 is scarce. This result was verified by Lonza through nanoscale flow cytometry. Similarly, we have found some unique marker sets in CDVs, which we have planned to analyze at the single particle level as well. Next, we examined these vesicles to understand how their unique properties might affect their potential as a drug carrier, first by looking at their shape. As you can see, it's nearly impossible to distinguish between these four circles. Although two of them are exosomes and the other two are CDVs, another exciting property of the CDVs is that they can be made into a powder and be quickly reconstituted by adding water while retaining their shape and biochemical properties. We also verified that vesicles maintain the original membrane topology of cells. Then we looked at individual nanovesicles to classify them with their surface markers. Initially, we used 3 classical exosome markers and found that approximately 12% of CDV particles contain those marker sets, compared to the much higher proportion typically observed from exosomes using a similar assay. This again indicates that CDVs have unique compositions compared to exosomes. We believe that the more unique marker panel for CDVs will further identify the entire subpopulations that constitute CDVs together. But whether they can be a good vehicle will ultimately be determined by their capability to load various cargoes or ability to deliver them to a specific location in our body. Over the past few months, we've been able to further improve the efficiency of RNA loading onto CDVs. Again, we confirmed that target genes are effectively suppressed when cells are treated with small RNA-loaded CDVs. When exosomes and CDVs are compared side by side, CDVs from 2 different cell sources showed similar or higher loading results than exosomes obtained from the same cells. On the other hand, by expressing membrane proteins on the surface of cells, we could obtain CDVs with a high level of target protein expression. And when cells deficient with this protein are treated with such CDVs, cells regain their lost function. Next, we added to cells CDVs or exosomes labeled with fluorescence dye. We found that recipient cells intake more CDVs than exosomes, as shown by flow cytometry or confocal imaging, while the intracellular trafficking pattern looks very similar over time between the two. We observed similar results from diverse cells. This even holds true for CDVs reconstituted from powder. We also found that RNA cargoes loaded with on the CDVs can enter these cells efficiently. In addition, when we injected CDVs into the eye, we found that CDVs can penetrate retinal tissues quite effectively and reach the epithelial barrier that has been a target for many debilitating eye diseases. Taken together, CDVs showed similar or better cellular uptake or tissue penetration results compared to the same number of exosomes. To summarize, we demonstrated that BioDrone has the potential to outperform exosomes as a drug carrier with its unique productivity that outnumbers natural exosomes. Along the way, we furthered our understanding to the finest single particle level, which will facilitate purification and quality control for large-scale manufacturing. Our next step is to prove the efficiency of our BioDrone platform at a more sophisticated level and demonstrate the functional consequences of such a BioDrones in various disease models. Finally, I would like to thank all the amazing members of MDimmune who have made this possible despite the challenges caused by the pandemic. I'd also like to thank all the project team members at Lonza for their excellent support, and of course, Plug and Play for making everything possible again. Thank you.